If you want to study mutations introduced by site-directed mutagenesis or variants of your gene, begin by determining which sequence is the longest version of your gene. You will need to order this sequence using the GeneArt Gene Synthesis service and set up the shorter versions as variants. Let's go to the GeneArt customer portal and get started. To set up the synthesis of the longest version of your gene, drag and drop the gene synthesis icon as shown. Then, select the biosafety requirements for your project. Next, work through the gene synthesis form by double-clicking the gene synthesis icon and entering the requested data and information. Note that if you choose to optimize your sequence using the GeneArt GeneOptimizer service, all variant sequences must be based on the optimized sequence. When you have completed the gene synthesis form, the status indicator should read Complete. To learn more about completing the gene synthesis form, please watch the Gene Synthesis tutorial. Now let's see the process for ordering multiple variants using the GeneArt portal. If you want to order several mutants or variants, drag and drop the variant bundle icon onto the gene synthesis icon as shown. The Variant Assistant opens. Choose whether to create variants using the parent sequence service or by uploading variant sequences using a multi-FASTA form. If you choose the parent service, the sequence used for the initial gene synthesis will be copied to the number of variant forms you specify. You can then introduce the variations you need into each variant form. If you choose the multi-FASTA form, you can paste all your variants into the system in one step. First, we will set up 2 variants using the multi-FASTA form. Choose the multi-FASTA option. The form expands, and you enter your prepared variants. Just copy and paste variant sequences into the sequence field in the portal. If you need extra services, select them here. Once completed, click Save. The program has created 2 single variants. Now open the first variant form by double-clicking. The name and sequence field are already filled in. You can change the name if you desire. In this example, we've chosen the name EPO variant. Finish completing the form as described in the single variant tutorial. Then click Save. The status indicator now reads Complete. Now complete the remaining variants in the same manner. Once the form for every variant is completed, you can proceed to the summary and send your project to GeneArt scientists for review. Now we'll go back and set up 2 variants using the parent service rather than the multi-FASTA method. So we're back to the step where gene synthesis for the longest version of the gene has been set up. Now drag and drop the variant bundle icon onto the gene synthesis icon as shown. The Variant Assistant opens. Choose the parent service option and type in the desired number of variants, 2 in this example, then click Save. The program has created 2 single variants. Open the first form by double-clicking. The name and sequence fields are already filled in. You can change the name if you wish. In this example, we've chosen the name EPO Variant 1. Now, type in your desired sequence modifications. Finish completing the form as described in the single variant tutorial. Then click Save. The status indicator now reads Complete. Now complete the remaining variants in the same manner. Once the form for every variant is completed, you can proceed to the summary and send your project to GeneArt scientists for review. Within 24 hours, the GeneArt team will review your project and send you an email notification. Then you can add the project to your cart and proceed to checkout. Please view the Add to Cart tutorial to see how that process works.